Good morning, everyone. My name is Mauro Cives, and I am Associate Professor of Medical Oncology at the University of Bari, Italy. First of all, let me express my gratitude to the INCA. It's truly a pleasure to be here to deliver this talk on the latest updates in translational net research. Transformative translational research is occurring in the NEN field. In particular, we have innovative research tools, innovative NEN models, and we have also innovative immunotherapy approaches. And in the last part of this presentation, we will discuss a little bit on that. Of course, it is impossible to have a compilation of all the new excitements going on in the field. So, of course, this is just a compilation of selected research. What are the innovative research tools that we have in the field? Well, for sure, single-cell and single-nuclei sequencing technologies will aid researchers in gaining a better understanding of the biology of neuroendocrine neoplasms. What is the main difference between bulk sequencing and single-cell or single-nuclei sequencing? Well, as you can see here, there is a clear difference, and this difference is that when you perform single-cell sequencing, you know exactly which cells are expressing what. And this is of utmost importance in NEN research, because in this way you may have a clearer understanding of what's going on. For example, this is a beautiful paper that was published a few weeks ago on Science by Hans Klever's group. And as you can see here in the right upper part of this image, in this, like when you see these colored dots, I mean, each of these dots correspond to a different cell. You can clearly see that when you're analyzing a tumor, you have different cells, and each of these cells is expressing different transcripts, different molecules of RNA. So, like basically, each cell has a different behavior. And, it is extremely important for us to understand which cell is expressing which marker. Why? Well, this is a paper that was recently published on iScience a few months ago. And like here you can clearly see, for example, that if you try to distinguish among NET cells in an individual patient, you can clearly see that you will have different fractions. But then if you merge the cells coming from different patients in a pooled sequencing analysis, you can clearly see that by single-cell sequencing, you will be able to dissect their behavior. See, for example, panel C. In yellow, you have all the cells coming from patients that are no metastatic, whereas in purple, you have all the cells coming from patients who had metastatic disease. And these are unsupervised analysis. That means that you get this analysis directly from the machine, and the machine is able to distinguish between the different cells. So why is this important? Because in the next features, we will be able to understand what are the differences, for example, between cells that are able to metastasize and cells that are not able to metastasize. And of course, by doing so, we will be able to understand what are the mechanisms leading to metastatization and eventually to fight them. But, for a long time we thought that the tumors are just a conglomerate of tumor cells, and that does not hold true at all. In tumors, we have a lot of other resident cells. There are healthy cells, for example, fibroblasts or endotheliocytes or immune cells and so on. And these sequencing studies are also shedding light on something that for us is completely new. Not only the tumor cells are different within the tumor, but also these tumor residing cells can be different, as you can see very clearly in these T3 panels. But, as our cities, tumors have their own organization. And it is crucial for us to understand better what is the special organization of tumors. I really like this picture because here on your left, you have an image from a tumor, and on the right, you have a city captured with a drone. And you can clearly see that there are analogies between how a tumor is built inside and how we build our cities. And of course, it is extremely important for us to understand what are the principles leading to this form of building the tumors, because then we can understand what are the vulnerabilities, and then we can tackle cancer. From this standpoint, nowadays we have new technologies that is called digital spatial transcriptomics that will definitely help us understanding what is the, let's say, spatial distribution of the different cells within a tumor, what are the relationship between different cells within a tumor, and why they are located in that area and not in another area. And of course, this will lead us to answer a very, very important question. Can we interfere with these mechanisms? And would this interference lead to better treatment? Not only, these new technologies also allow us to understand what is the expression of tumor markers, specific tumor markers, for example, in this case, messenger RNAs, in selected group of cells. So if you are interested in a certain population of cells, then you can go there on the spatial distribution of the tumor, and then you can get the insights from exactly the type of cells that you're interested in. But not only we have new research tools, we also have innovative NEN models, and this is of course extremely important for finding a cure for NENs. In particular, last year, Talia Dayton and colleagues published an extremely interesting work where they were able to establish for the first time patient-derived tumor organoids. What are these patient-derived tumor organoids? So basically, they were able to grow the neuroendocrine tumors in a plate. And these tumors had the same identical characteristics of the tumor in the patient. Is this important? This is of utmost importance. And the reason for that is that, of course, we can use this model to predict, for example, the response to drugs. Nowadays, we do not have predictive factors for all the treatment that we have in the field of neuroendocrine neoplasms. And of course, the only thing that doctors can say to the patients is that we need to try this treatment, and then we will see if this treatment is going to work. By exploiting these new models in the next future, we will be able to test treatments in vitro before we administer that specific treatment to the patient. And for example, in this work, you can clearly see that there is a tight correspondence. The red dots are indicate that there is a high sensitivity in vitro to a certain drug, whereas here you see the clinical response. So for example, where you see the red dots, these patients then they underwent partial response. That means that they had a good response to the treatment. Whereas when you see the blue dots, that means that in vitro there was a poor response to the treatment, and indeed those patients got progressive disease. That means that they did not respond to the treatment. This proves that we may be able in the future to tailor our treatment according to preclinical knowledge. Precision-cut tumor slices are another innovative model that we're going to use in the next future. In essence, in precision-cut tumor slices, you cut the tumor when the tumor is still viable, and then you are able to incubate the tumor and to have the tumor living for up to 7-10 days. And then you can basically perform experiments on these slices For example, you can test drugs. You can see what are the effects of these drugs on the tumor, or you can try to understand what is the biology leading the growth of the tumor. And as you can see in this picture, the precision cut tumor slice, the part on the right part of this table, of this figure, resemble the clinical histology of these patients. Again, it is impossible in 20 minutes to deliver a talk encompassing all the new advancements in the NEN field. So I decided to exclusively discuss advances in immunotherapy in the field of neuroendocrine neoplasms. In particular, we're facing a moment in which we have a lot of excitement around immunotherapy. As you may know very well, immunotherapy with immune checkpoint inhibitors has shown a limited activity in neuroendocrine neoplasms. So of course we need to be bold and we need to explore novel strategies to fight the cancer through our immune system. And for example, this is something that was explored last year, where, like, for example, they tested whether the combination or the sequencing of PRRT and immunotherapy could lead to better results in mice models of neuroendocrine neoplasm. And in particular, what they showed is that when you deliver PRRT first and then immunotherapy, you may have better results in terms of tumor shrinkage in the mouse model. And as you can see in these beautiful images, the red spots are T cells, that means lymphocytes, T lymphocytes, that are expressing molecules that are able to kill the tumor. Its name is granzyme B. These are molecules that kill the tumor cells. And you can clearly see that when, like here in the red part of the image, when you have the sequence PRT first and then immunotherapy, you have way more production of granzyme B by T cells. And of course, that aligns with our expectation in terms of sequencing. And when you test the effect of this sequence, PRT first and then immunotherapy, in the murine model, you can clearly see that when you have the sequence PRT and then immunotherapy, you can have basically the eradication of the tumors in the murine model. This concept has been tested in the context of a clinical trial, but the results have been not that exciting as the preclinical results. This was a cohort of patients mostly comprising small cell lung cancer patients, so maybe this is not the perfect cohort to test this concept, but anyhow, there are studies that are now addressing these research questions, and we will hear about it very soon. Antibody-drug conjugates are an emerging class of drugs in oncology. Different substances can be linked to antibodies, and in particular, if you link a toxic substance to an antibody, you will be able to specifically bring this poison within a tumor cell. And that's what we do with this class of drug that we call antibody-drug conjugates. Several of these drugs have been developed. There is a study by a colleague at the University of Alabama at Birmingham that developed this compound, and there are also other compounds. For example, this is being developed by Crinetics, and this is a non-peptide drug conjugate. So in essence, this drug is able to target somatostatin receptors. Then the compound is internalized, so it goes into the cell. And once into the cell, this compound is able to kill the cell because, again, there is this poison that is attached to this monoclonal antibody, and this poison is going to kill the tumor cell. There are also other antibody-drug conjugates that are currently being investigated in the neuroendocrine field. In particular, there is another protein whose name is DLK1, which is well expressed in pheochromocytomas and paragangliomas. And as you can see here in the right part of this slide, there is a compound that is able to eradicate tumors in the murine model when the tumor cells express this specific protein. Now, there is a phase 1 dose escalation study up and running at NIH in Bethesda. Adopted T-cell therapies have sparked an enormous interest in general in oncology, but also in the NEN field. In particular, research exploiting CAR T-cells or TILs has reached the clinical approval. CAR T-cells are engineered T-cells, engineered T lymphocytes, that are basically able to attack in a very specific way the tumor cells. Different CAR T-cell products are currently approved in hematology, in particular for the treatment of different forms of leukemia and lymphoma. For sure, it's more difficult to tackle solid tumors by exploiting CAR T-cells, but a lot of investigations are trying to move forward the field. Different generations of CARs stand for chimeric antigen receptor, have been generated so far. And what I would like to highlight is that the so-called next generation of CAR T-cells is also able not only to redirect the specificity of T-cells against tumor cells, but also is able to lead to the secretion of specific molecules by the T-cells themselves when they encounter the antigen, namely the protein that makes the tumor cell be recognizable by these lymphocytes. How does the system work? So basically you start with an apheresis, so you take the blood from the patient, then you isolate the T cells. The T cells are then engineered, and then they are infused back into the patient. And then they are able to circulate to find the right tumor cells and to kill them. Several CAR T cells have been established in the NEN field so far. In particular, anti-CD817 CAR T cells have been developed by colleagues at UPenn. We developed in collaboration with colleagues at Moffitt Cancer Center for T cells targeting somatostatin receptors. There are several academic and industry CAR T cell products targeting DLL3 and other protein extremely important, in particular in high-grade neuroendocrine neoplasms. And then there is another CAR T cell product targeting interleukin-13 receptor alpha 2. In particular, what I would like to highlight is that all the preclinical research is already translated into clinical research, and phase 1 clinical trials are currently up and running, for example, for the anti-CD817 CAR T cell product, or for the anti-IL-13 receptor alpha 2 CAR T cell product. So there are clinical trials testing for T cells in the field of neuroendocrine neoplasms. A few months ago, FDA approved a drug named lifileucel for the treatment of melanoma patients who progressed on immune checkpoint blockade or targeted therapies. What is lifileucel? Lifileucel is a product made by tumor-infiltrating lymphocytes. So basically you take the lymphocytes out of the tumor, you grow them in a lab, and then you reinfuse them back into the patient. Why so? Because if the lymphocytes were within the tumor, they may be able to attack and kill the tumor cells. And if we are able to grow this army of soldiers, then we will be able to attack the tumor. And currently, there are studies including our study funded by NET Research Foundation, that are trying to understand if treatment with the tumor-infiltrating lymphocytes can be used for the treatment of patients with neuroendocrine neoplasms. An emerging class of agents in oncology is the class of BiTEs. BiTEs are bispecific T-cell engagers. So basically these are molecules that are able to put in close contact, as you can see here, the lymphocytes, the blue cell here, with the cancer cell, the pink cell that you can see here. And once they put these cells in close contact, they are able to activate lymphocytes so that they're able to kill the tumor cells. Tarlatamab is a first-in-class BiTE, and it has been approved for the treatment of patients with the previously treated small cell lung cancer. And this is a type of neuroendocrine neoplasm, as you know. And currently, there are clinical trials assessing other BiTEs targeting DLL3. We talked about this protein a few minutes ago in patients with high-grade neuroendocrine neoplasms. There is another product targeting the same protein. Not only, viral therapy is another potential way to treat neuroendocrine neoplasms. A few years ago, a viral product was developed. In particular, they were able to develop this virus that was able to target specifically neuroendocrine neoplasm cells. And within the virus, there was a substance that is toxic for the cell. So basically, they were exploiting this virus to bring, again, a poison within the tumor cell. And in these beautiful images, you can see that the green spots. The virus went only to the right cells, to the tumor cells. It didn't go to other cells in the body, for the brain, heart, muscles, spleen, kidney, and so on. But most importantly, investigators were able to demonstrate, as you can see here, this is a CT scan of a mouse before and after the treatment. They were able to demonstrate that the virus was able to reduce the size of the tumors, and in particular, to prolong the survival of the mice. Vaccines. Prophylactic and therapeutic cancer vaccines are being currently investigated in oncology and also in the field of neuroendocrine neoplasms. You may all know how vaccines work, and the principle of cancer vaccine is identical to the vaccine that we use, for example, for the flu. So you have a vaccine injection, then this vaccine will stimulate specific immune cells to recognize antigens, small proteins, small, let's say, markers that will render these immune cells able to attack and kill the tumor cells. Sorvevin is a protein expressed in many cells, including neuroendocrine neoplasm cells, and colleagues at the Roswell Park have developed a vaccine targeting this protein, which again is overexpressed in neuroendocrine neoplasms. And currently, there is a clinical trial investigating this product in patients with neuroendocrine tumors. Not only, we previously demonstrated that neuroendocrine neoplasms, in particular pancreatic neuroendocrine neoplasms, do express neoantigens, namely small proteins that can be recognized naturally by immune cells. Look what happened a few months ago in the context of another cancer. This is hepatocellular carcinoma. They were able to develop personalized neoantigen-based vaccines. So basically, you recognize what are the difference between the tumor and the healthy cells, and then you create a vaccine emphasizing these differences so that the immune system will be able to understand which are the bad cells. And then the immune system will be able to recognize them and to kill them. And as you can see here In this, we call them as spider plots. Each line is a different patient. You can see that, like all the blue lines mean that the patient, they were really responding very well to the treatment. The tumor was shrinking. And you can see that the responses were also long-lasting. New research tools to conclude, for example, single-cell sequencing or spatial molecular profiling and new NEN models will definitely help researchers to gain a better understanding of NEN biology and, of course, to understand in particular what are their vulnerabilities in the near future. We definitely need transformative translational research because this is the only way we will be able to improve patients' outcomes and eventually cure NENs, and we need to be bold. But an alliance must be built between patients advocates, researchers, and clinicians. We do not need, let's say, science for science, like heart or l'art pour l'art. We need transformative, bench to bedside, very focused research that will be able to advance the field, not in 100 years, but in the next decade. To do so, scientific questions must be discussed with patients and patient advocates. And in particular, for example, patients' advocacy group may have a role in grant funding. And preclinical research must be translating in clinical research. There are a lot of preclinical products that will never reach the clinical testing phase. And of course, these are all products that hold a potential to transform our field. That means that we need to reduce the so-called attrition rate, and this must be a public priority. With that, I would like to thank you very much, and see you very soon.